The cell has three main components. The outer boundary is called the cell membrane. Typically, there's at least one central structure, and that's called the nucleus, which contains the genetic material. And then there's the stuff between the plasma membrane and the nucleus, and that is the cytoplasm, which in turn contains a background semi-fluid gel area that is called the cytosol. Within the cytosol are floating around two types of things: organelles. An example would be mitochondria. These are permanent structures within the cytoplasm. And then there are inclusions, which are temporary components, including things like lipid droplets, fat droplets, melanin, which is the pigment skin cells produce. Glycogen would be the storage form of glucose. First, we're going to take a look at the plasma membrane, also known as the cell membrane or plasmalemma. In this diagrammatic, you could see that within the plasma membrane are two layers of phospholipids, which contain a polar hydrophilic or water-loving structure, which faces the external and internal liquid environments, and a nonpolar hydrophobic or water-hating area, and that faces towards the interior of the plasma membrane. There's also proteins. Proteins are chains of amino acids. Two types of proteins here: integral and peripheral. Peripheral proteins would be either on the outer or inner region of the plasma membrane, whereas integral proteins are mostly transmembrane, which means they go straight through the two phospholipid layers, and many of them have internal channels. Then there are cholesterol molecules to stabilize the integrity of the membrane. And finally, a glycocalyx, which is the outer sugar coat biological marker of each cell. And the glycocalyx includes glycolipids, sugars to fats, and glycoproteins, sugar to proteins. Movements across membranes. Structures, molecules, small or large, need to get through the membrane. Different types of mechanisms by which this occurs. If you are currently taking physiology, you'll learn much more about this. Simple diffusion is where small molecules, for example, oxygen molecules, could zoom right through the membrane, going from a higher to a lower concentration, that is to say, down their gradient. Osmosis is nothing more than water molecules diffusing across a membrane. It could be either into the membrane or from the cell out. Osmosis, diffusion of water molecules. Then we're simplifying things in this class. We will give the term transport mechanisms to define any movement across a membrane where transport proteins as carriers or pumps are used. Transport mechanisms utilizes protein carriers or pumps. Vesicular or bulk transport. That means getting larger chunks of stuff into the cell or from the cell out. Endocytosis, into the cell, The opposite would be exocytosis, which is in the next slide. Phagocytosis, cell eating. This happens when, let's say, white blood cells want to engulf a solid particle, could be a bacterial cell, in which case they throw out these false feet or pseudopods, engulf the solid particle, and internalize it as a phagosome or eating body. The content of the phagosomes is usually broken down by a digestive type organelle known as a lysosome. Pinocytosis is cell drinking. The cell internalizes in vesicular form. A vesicle is a little bubble defined by the membrane. Cell drinking or pinocytosis is a process that most cells can undergo. The most specific type of internalization of specific molecules is known as receptor-mediated endocytosis. And here, protein receptors are fishing around for a very specific cargo of molecules. Once those receptors are filled in, the entire bubble or vesicle pinches off, and this now newly formed vesicle contains a very specific cargo of molecules. Exocytosis is just the reverse of endocytosis, from the cell out. Organelles, this is something you're probably familiar with, but let's go at it anyway. Mitochondria would be plural, one mitochondrion, are double membraned structures that include an inner infolding. Those are called cristae. Mitochondria form ATP, adenosine triphosphate, a high energy yield molecule. Mitochondria are the powerhouses of the cell. Lysosomes, we already talked about, contain enzymes known as acid hydrolases, and lysosomes digest material. Rough and smooth ER. Rough ER is referred to as rough because it contains or is studded with ribosomes. Rough ER works in protein synthesis. Smooth ER or SER functions to detoxify some structures, also in lipid or fat metabolism, and especially in muscle cells, the SER serves as a calcium storage organelle. Centrioles are composed of microtubules. We'll see later that microtubules form the mitotic spindle during cell division. Golgi apparatus packages proteins. It receives proteins from the rough ER, repackages them, and perhaps sends them out in the form of exocytosis. In this picture, you could also see microtubules, which are not only internal structures of centrioles, but also form the cytoskeleton, microscopic skeleton within the cytosol. Peroxisomes are more specific to liver and kidney cells. They contain catalases and oxidases. If that doesn't sound familiar to you, if you haven't had chemistry, don't worry about it. But please know peroxisomes clear free radicals. Free radicals are byproducts of chemical reactions that could cause harm to the cell. Again, peroxisomes are more specific to liver cells and kidney cells. Ribosomes attach to the ER, endoplasmic reticulum, typically. They are made in the nucleoli. Each ribosome consists of two subunits. Each subunit is made up of ribosomal RNA and protein. Ribosomes become attached to a string known as the messenger RNA. Messenger RNA is a copy of DNA, and it contains the code for specific proteins. You'll learn protein synthesis in both chem and physiology. Taking a closer look at the ER, endoplasmic reticulum, again we have the smooth and the rough ER. One more look at the mitochondria, double-layered membranes, organelles. Internal membrane is thrown into cristae. Those are crest-like structures. And mitochondria synthesize ATP. Another look at the Golgi. Golgi receives proteins from the rough ER, packages these proteins into new vesicles, typically for secretion. Next slide shows the various processes where proteins formed on the ER are then transferred to the Golgi where they are repackaged. What happens at that point depends on where the proteins are going. They're either exocytosed, or if they are full of acid hydrolases, this pinched off vesicle then becomes a lysosome. Centrioles usually appear in pairs. Again, they're composed of microtubules, and they form the mitotic spindle during cell division. Cytoskeleton, miniature little tubes within the cytosol. We mentioned microtubules already. Those are the largest. The smallest are microfilaments. An example would be actin. If you have taken physiology before or any muscle studies, you know that actin is a contractile type of a protein. And then finally, we have intermediate filaments, intermediate in size between microtubules and microfilaments. Intermediate filaments provide for strength, tensile strength for the cell. Nucleus. The nucleus is defined or protected by two membranes. Together, they are referred to as the nuclear envelope, which has tiny little pores for things to get in and out of. The material inside a nucleus of a cell that is not dividing contains chromatin, which is basically DNA and protein. The nucleoli would be one nucleolus, plural nucleoli, are the darker structures, more condensed in a nucleus, and nucleoli are the ribosome factories. Cell cycle. This is defined as the life cycle of a cell. A cell that's going to divide will have not only interphase, the regular growth, replication of DNA and centrioles kind of phase, but also cell division. And in cell division, we divide not only the nuclear content, that's known as mitosis, but also the cytoplasmic content, and that is known as cytokinesis. In this diagrammatic, you could see that the initial parent cell, with its 23 pairs of chromosomes, has to duplicate those chromosomes or chromatin, and also the centrioles duplicate or replicate prior to cell division. All of this preparation happens in interphase. Then again, when the cell undergoes division, it has two parts to that cell division, mitosis and cytokinesis. Another picture of this interphase versus the beginning of cell division. In interphase, what happens? In the nucleus, we see chromatin, DNA in the form of chromatin. But towards the end of interphase, what happens? Both DNA doubles or replicates and centrioles replicate. Now, once a cell starts to undergo cell division, it goes into the first phase of cell division, which is prophase. The last slide of this chapter includes a diagrammatic of the different phases of cell division, mitosis basically. The different phases are from beginning to end: prophase, metaphase, anaphase, and telophase. I came up with the same, which is not great, but if you remember, please make another 2 cells. The P in please would be the P in prophase, M in make, M in metaphase, if that helps at all. So what happens? In prophase, the chromosomes appear, the nucleus and the nucleoli dissolve, and the centriole pairs migrate to the opposite ends of the parent cell. And as they do so, they form the mitotic spindle. Metaphase. Remember M for meta, M for middle. What happens in metaphase? All the chromosomes line up in the middle. Meta, middle. Anaphase, ana means apart. During anaphase, the chromosomes pull apart and start pulling into the now starting to form newly formed daughter cells. Telophase. If you remember what happened in prophase, you could just reverse it, because in telophase, just the opposite happens. Chromosomes disappear, the nucleus and nucleoli reappear, and the centrioles are no longer making or forming the mitotic spindle. Cytokinesis, the unequal division of cytoplasmic content, typically starts during late anaphase and is definitely completed by the end of telophase.